חברים, יד הגורל האכזרי הביאה אותי בפניכם היום. אתם יודעים שהיושב ראש של ישיבת אחרי הצהריים היה צריך להיות פרופסור אלסברג ברגמן, אבל הוא נלקח מאיתנו לפני כמה שעות. הייתי מבקש מכם לקבל את זכרו בקימה. תודה. קשריו של ארנסט דוד ברגמן עם המכון ועם המדע בישראל ועם ביטחון ישראל ידוע לכולכם, ועוד נמשיך ונרבה לספר בפעילותו בימים הבאים. אני רק רציתי להגיד כמה מילים אימפרומפטו, כמו שאומרים, בלי הכנה מוקדמת, ובלי שזה מהווה סקירה מקיפה על חייו ופועלו של ארנסט דוד במכון. התלבטנו גם אם לקיים את המעמד של היום או לבטלו, כי בכל אופן ברגמן לא היה סתם יושב ראש של ישיבת אחרי הצהריים ולא סתם אחד מבדלני המכון, כי הוא היה משהו מיוחד במינו בכל ההיסטוריה של המדע בישראל. אחרי מחשבה נוספת, הרגשת כולנו הייתה שבסך הכל ברגמן עצמו היה רוצה שהמדע ימשיך ויתקדם למרות זה שהוא איננו איתנו. מתוך הרגשה כזו הרגשנו שנבונו יהיה להמשיך בסדרת ההרצאות שמתחילים אותה היום ולחזור ולהתייחד עם זכרו של ברגמן במאורע מיוחד, ועוד תהיינה לנו הזדמנויות על כך. דבר אחד אמנם ביטלנו, וזה את קבלת הפנים של אחרי ההרצאה הזו, ואני בטוח שאתם מבינים את המניעים לכך. ברגמן הצטרף למכון בשנות ה-30 המוקדמות. לא עשיתי עוד מחקר מדויק, לא הספקתי אם זה היה ב-34 או 35 או 33 וחצי. בכל מקרה, הוא היה בין האנשים הראשונים שפרופסור ויצמן הביא לארץ להקים את המוסד המדעי המחקרי הראשון. זה גם יד הגורל שאנחנו מאחדים את שני השמות האלה היום אחרי הצהריים, את שמו של ויצמן ואת שמו של ברגמן. כי כברה ארוכה עברו שניהם בארץ. כפי שאמרתי, מ-33, שברגמן כהן כמנהל הראשון של המכון זיו, ואחר כך מכון ויצמן, עד מותו של ויצמן, הם למעשה עבדו כמעט בכפיפה אחת, פרט לכמה בעיות בסוף השנים. ולכן חלק גדול מאוד יש לברגמן בעיצוב דמות המכון, בעיצוב דמות המדע בכלל, בעיצוב השיתוף בין מדע ומדינה, מדע וביטחון, ולכל אלה תרומתו נכבדה ויחידה במינה. אינני יכול ברפרוף ראשון להזכיר אדם אחר מחוגי המדענים בארץ שיש לו זכות כה גדולה לתרום לקידום המדע והתפתחות הארץ. כאמור, נמשיך היום לפי התוכנית. נהפוך את ההרצאה הזו במידה מסוימת להרצאת זיכרון משותפת לויצמן ולברגמן. אנחנו מתחילים בסדרה של 3 הרצאות שהן הרצאות הזיכרון על שם חיים ויצמן, שהן מתקיימות כל שנה. והשנה, כפי שאתם יודעים, הרצאת הפתיחה ניתנת על ידי נשיא המדינה, שגם הוא אחד ממדעני הראשונים של המדינה, של מכון ויצמן, של מערכת הביטחון, וגם הוא בעצם דרכו חפפה שנים רבות את דרכו של ברגמן. שתי הרצאות אחרות תינתנה על ידי הנס קורנברג ותינתנה בזמן שנקבע. ובכן ברשותכם אני אבקש עכשיו את מרצה הראשון, את פרופסור אפרים קציר, אליו לשאת את דבריו. כפי ששמתם לב, אני לא מציג אותו בפניכם, נדמה לי שזה מיותר לגמרי. בבקשה.
ישראל, ידידים, במידה מסוימת קשה לכולנו להמשיך היום בתוכנית הרגילה, ולי קשה הדבר במיוחד, משום שאני נמנה על המספר הלא גדול של חברים שהייתה להם הזכות לעבוד עם ברגמן הרבה שנים, להיות איתו במכון ויצמן בשעה שהוא ניהל את המכון ועיצב את דמותו של המכון, לפעול איתו ביחד בחמ"ד, אחר כך ברפאל, אחרי כן באקדמיה, ואחרי כן בעניינים לאומיים אלה ואחרים, כולל האוניברסיטה העברית. לכן קשה המלאכה עליי היום. הייתי רוצה את הדברים שאומר במידה מרובה להקדיש לזכרו של ברגמן. ברגמן אשר תמיד יישאר לי כדמות אולי של המדען הדגול ביותר שמדינת ישראל הקימה, מדען אשר מסירותו לעמו לא ידעה גבול, ועשה כן עד יומו האחרון. עוד לפני יומיים שוחחתי איתו טלפונית. הוא אמר לי שהוא מרגיש מאוד לא טוב, הוא נכנס לבית החולים, אבל הוא עושה זאת לא למען בריאותו, כדאי ירבו לספר על פועלו, על האיש, וטוב שימשיך לשמש לנו דוגמה. כאשר נתבקשתי, אמשיך בהרצאתי באנגלית, ואנסה לזכור כמה מן הנושאים אשר ביקשתי להעלות. I will continue and discuss my scientific topic in English as I have been asked to do. The topic has to do with enzymes, fermentation, and biotechnology, and I would perhaps like to introduce some of the work and some of the problems raised as a result of the group which has been working and is still working at the Weizmann Institute and has perhaps indirectly some connection and ties with the work of Professor Weizmann. I'm quite certain that all of you are aware of the fact that already in 1912 and even before, Weizmann was engaged in fermentation problems, in anaerobic fermentation, and he discovered a bacteria, a Clostridium acetobutylicum, which is named after Weizmann, which can produce on fermentation, on anaerobic fermentation, acetone and butanol. And sometimes one wonders, why did a chemist and an excellent organic chemist like Weizmann suddenly turned to a bioengineering, biotechnological process. He was a superb chemist. And it seems to me that the real reason was that Weizmann was very much interested in synthetic rubber at that period, and he was looking for new sources of isoprene in order to produce, by a polymerization technique, synthetic rubber. And after a short while, he reached the conclusion that via fermentation, it will be possible, perhaps, to get some 5 carbon compounds from which he will be able to produce isoprene. This is the reason that he looked already at that time for a fermentation process, and this is the reason that his boss in Manchester, when he discovered that this Clostridium acetobutylicum produces acetone and butanol with 4 carbons and not with 5, he thought that Weizmann is really wasting his time, and his recommendation was to throw out this little bug as quickly as possible and to return to good organic chemistry. But Weizmann, as we all know, continued, and as a result of this work, he finally established a process, one of the first processes, anaerobic fermentations, in which products of industrial value have been obtained. This process he developed during the First World War, and it's rather interesting that he found out that the Clostridium that he used is capable to utilize starch, molasses, starch from corn and other sources, break it down finally to simple sugars, and by anaerobic fermentation from 6% sugar to produce about 2% of weight by volume mixed solvents composed of about 65% butanol and 30% acetone and 5% ethanol, and in addition CO2 and hydrogen. Now, as is well known, the acetone became of great importance during the Second World War as a solvent for cordite, and after the Second World War, butanol, mainly in the form of esters, became an important solvent for various polymers and lacquers to cover vehicles, cars, etc. So it was a most successful process which has been used for many years. But it's rather interesting that when one reads Weizmann's patents and Weizmann's thinking about these fermentation processes, one immediately realizes that in addition of this simple fermentation product that he studied very, very carefully, and there are very many patents dealing with this process, he had a kind of a general outlook. And he thought that perhaps time will come when fermentation products will be a source for many new compounds to be synthesized. He even went so far as to mention that some of these products might serve as a source of energy. And his idea was, look, carbohydrates are products of photosynthesis by which one utilizes solar energy. But most of the carbohydrates are solid materials, and if you want to form a liquid, you just carry out a fermentation process, and you have a liquid which is to a certain extent energy rich. Moreover, he thought that some of these materials will be of great use as source for the growth of yeast and thus produce cheap proteins, a topic that has been recently pursued very actively in all the experiments to get single-cell proteins on petrol, methanol, methane, and other materials. When I looked up the literature about fermentation, I was enchanted by the fact that somehow similar ideas to those that Weizmann had, of course Pasteur had, when he started his work on fermentation. Pasteur, as all of you know, started as a good chemist, a chemist who mainly wanted to please his teacher Biot, who, by finding asymmetric molecules and finding molecules that will rotate polarized light, he thought that he has perhaps achieved one of the most important achievements in his life. But as a very young man of 33, Pasteur turned to biological fermentations as a chemist. And he did so because he was very much interested in asymmetric molecules, and he found out soon that amyl alcohol, which is found in fermentation, is asymmetric, and therefore via asymmetry he went to fermentation. And of course in Lille, where he has spent his work on fermentation, he carried out his classical work on fermentation, which he published in 1857. And it was the first proof that fermentation, it was lactic acid fermentation, is caused by bacteria, by living organisms, and afterwards he of course extended this work very much to very many other types of fermentations. But as we recall today Ernst Bergman, I think that Pasteur and Weizmann had perhaps in their research the same type of motives to help the society in which they live. And it's rather interesting that just half a year before the publication of this paper of Pasteur in 1857 in Lille, Pasteur called in all the professors of his institute, and he told them the following: "Look, gentlemen, we live in the northern part of France. We have to help industry and agriculture in this place, and it has to do with fermentation and sugar, mainly sugar from beets. We have to do something about it." This is how he started to study alcoholic fermentation, and one of the big producers of alcohol found out that here and there his fermentation doesn't go well and acid is formed. This is how he reached the conclusion that something is wrong, and he looked in this wrong acid ferment, and he found out these lactic acid bacteria which produce lactic acid, and he reached this wonderful conclusion about fermentation in general. And his first advice to the producers of alcohol was, look under the microscope for this little bacteria. If you find it, stop fermentation. It will lead to lactic acid. Look for yeast. They will give you good alcohol. But this work that Pasteur started in 1857 didn't continue very actively, and Weizmann was one of the first who in 1912, after more than 50 years, started for the first time to use fermentation for the production of an industrial group of compounds. Of course, during the First World War, Neuberg produced this interesting method to form glycerol by fermentation, and large-scale products have started at this period. But still, biochemical engineering wasn't in existence. And it is my feeling that this important branch of science, in which engineers and biologists participate together, has really started with during Second World War, when antibiotics and vitamins have been, production of which has been initiated by microorganisms. Of course, the story of penicillin production is so well known that one doesn't have to survey it at this point. But of course, there all the problems in which a chemical engineer is faced have appeared. Slowly from that time, from Second World War, the biochemical engineer has been involved in many characteristic problems of a bioengineer, in fermentation processes, aerobic and anaerobic, in food processing, pasteurization, sterilization, preservation, freezing, deep freezing, cooking and drying. All of these are new arts of the modern bioengineer. Manufacture of saline vaccines, extraction processes, physical processes of clothing materials, cotton, wool, processing of forest and crop products, and adequate equipment. All these are by now producing a new branch of science and engineering, which is a rather interesting branch of science. Of course, the engineers are trying to divide their branch of research and study into unit processes of heat transfer, of agitation, of aeration, of diffusion, whereas the biologist, of course, drives more and more to study the biological processes. But I have chosen this survey in order to suggest that now we are entering in the area of bioengineering a new period, a period in which the bioengineer will utilize more and more, not living organisms, microorganisms, but catalysts which are derived from these microorganisms. And what I have in mind are enzymes which can be derived from microorganisms and be used in order to carry out various catalytic reactions and processes. It is perhaps worthwhile to mention that since the times of the crystallization of urease in 1926, a lot of information has been acquired about these catalysts up to the primary sequence of lysozyme, Stein and Moore, and the three-dimensional structure of lysozyme of ribonuclease by Stein and Moore, and lysozyme, three-dimensional structure by Phillips. Altogether, the biochemist deals with about 1,500 enzymes. It is said that most likely in nature we have tens of thousands of enzymes and maybe even more. Catalysts, very specific ones. It's rather strange to realize that out of this enormous number of enzymes, only about 100 or 150 of these enzymes have been investigated rather thoroughly. All the rest are really not known well. But when one thinks about the possible use of these catalysts and the advantage in their use in comparison with microorganisms, it's perhaps worthwhile to mention at least a few. Microorganisms, during their growth, undergo a cycle, and during this cycle, of course, they change in their metabolic processes. So once you grow bacteria or yeast or mold or fungi, they will change their metabolism, the producing of certain materials during growth. They are composed of a large number of catalysts, and when you grow them, naturally many processes occur simultaneously. The fact that they grow leads to clogging and all the characteristic problems which are connected with growth, whereas if you'll have enzymes and if they are being used as catalysts, you can even pick up very characteristic enzymes which will carry out reactions which undergo in the cell only as a sideline, and to transform these catalysts into the main process in your reaction. You can certainly combine enzymes and thus carry out a complex set of well-planned reactions. Now, the modern enzyme engineer says that enzymes will be used more and more in diagnostic testing, in replacing high temperature and pressure, in developing new food sources from cellulose, petroleum, other materials, in developing new processes for anti-pollution techniques, in producing new sugars, producing new sources of energy, and being used in order to utilize solar energy. Some of them are going so far as to suggest that batteries and flow of electrons will be produced by enzymes, as in a few cases, at least it has been shown already experimentally. And last but not least, I assume that within the coming near future, some enzyme electrodes will appear on the market with which one will be able to determine specifically the nature and concentration of various materials in biological and other media. Now, I have mentioned this area of enzyme engineering, which I feel is perhaps a branch of modern bioengineering and of modern fermentation, because of my interest in this area and the work that has been carried out in this area by a number of collaborators at the Weizmann Institute, by Rachel Goldman and Silman and Yuda Levin and others, who have tried to transform enzymes into something that can be utilized in industry in an easier way. And I'll perhaps survey some of the previous work, indicate some of the recent developments, and emphasize some of the major problems that a modern enzyme engineer has to cope with. Now, our first idea was that because of the fact that so many enzymes in biological systems appear in membranes or appear in bound systems, it will be worthwhile to bind enzymes to various carriers and to investigate the effect of the environment, the carrier, on the enzyme. Of course, we are well aware of the enzymes of the respiratory system enzymes participating in transport, in protein biosynthesis, in photosynthesis, all of them appear in membranes or more complex structures. We thought that if one can bind enzymes to various carriers, these enzyme carriers, if these carriers are inorganic or organic, these carriers will be particles which will be handled rather easily, and one will be able to use these particles for columns, for packing columns as enzymes, or to produce membranes. So the idea was a rather very simple one. But in order to tell you what has been done just already to start with this technique of binding enzymes to various carriers, and we thought mainly covalent binding via non-essential groups of the enzyme under very mild conditions which will not denature the enzyme, I have picked up just a recent article about an immobilized lactase, and the author has summarized the various carriers to which immobilized lactase, which splits lactose, this sugar of milk, what were the various carriers that various authors have succeeded to bind this enzyme. Now let me start by glass. You can bind this enzyme to glass and get beads with enzyme attached covalently to it. Ceramics, zirconium, aluminium, steel. You can get now particles of steel to which enzymes are attached. You can certainly have polysaccharides like cephalodex and cellulose and agarose. You can have collagen fibers. You can have nylon particles and fibers. I hardly wonder whether there is anything in this world to which one cannot bind now covalently an enzyme. But there are very many techniques that one has to face and many problems that one has to ask oneself once you are going to attach an enzyme to a carrier. Now, in order to just to survey very briefly the older literature, I have really picked up here a few examples and added some new examples in order to show you the sophistication by which an organic chemist now can use in order to produce such immobilized enzymes. And here we'll... Let's have a look at the first slide. Now here you have one of the oldest reactions by which one can bind to carboxymethyl cellulose various proteins and enzymes. Here you have carboxymethyl cellulose, you make an ester via hydroxide, you make a hydroxide, hydrazine, you make an azide, and of course, if the amino group of the protein is not essential, alpha or epsilon, you can couple it under relatively mild conditions, and you have particles or fibers of cellulose with protein. Next slide. Here you have, this is a very well-known reaction which Dr. Porat has worked out in Stockholm. It's a bromocyanogen reaction with polysaccharides. He uses mainly agarose or saccharose or cephalodex. If you treat it with bromocyanogen, you end up with an aminocarbonic acid, and the aminocarbonic acid will react with the amino group of the protein. You end up with this intermediate derivative which finally will give a carbonic acid which is very stable. It's a very, very commonly used now method to bind antibodies, protein antigens, enzymes, etc., if the amino group is not essential. Next slide. Well, here is an old copolymer that Yuda Levin in our laboratory used. It's an ethylene maleic anhydride copolymer. If you treat this copolymer with trypsin, for example, the amino groups will react with the maleic anhydride. You'll have it cross-linked, and of course those maleic anhydride residues which have not reacted will open up, and you have a anionic type of a polymer surrounding the trypsin. Next slide. This is a rather sophisticated carrier that Leon Goldstein enjoyed. If you take a polysaccharide, you can take starch or any other polysaccharide. You oxidize it with periodide, you get a dialdehyde here, and if now you cross-link the dialdehyde with hexamethylenediamine, you'll cross-link it. Of course, you'll get the Schiff base. You can reduce the bond with sodium borohydrate. And if you add here a diaminobenzidine, then you'll have compounds which you can diazotize. And if you'll diazotize, you can use this link to combine enzymes via their tyrosine or other aromatic groups. So this will you'll end up with a polymer with many basic groups if you'll bind here a protein. Next slide. This is strictly a hydrophobic copolymer. It's a schematic leucine and diazotized para-aminophenylalanine to which an abstract papain has been adsorbed. So this is an abstract painting of papain, the active site. This is the tyrosine attached. It's yellow, of course, and it's a very stable enzyme. Bound to this leucine para-aminophenylalanine copolymer, which is completely water insoluble. Next slide. Here is another technique by which one can cross-link molecules of enzyme by bivalent reagents, like glutaraldehyde, this diisothiocyanate, this difluoride. Here you have a trichlorotriazine, and other bivalent or trivalent molecules. They will bind very many enzymes one to another, and you have particles of enzyme. Next slide. But here come a little bit more sophisticated techniques, and one technique which I want to mention rather briefly of Fukui and his collaborators in Tokyo, work that they have published just a couple of months ago, is based on using cofactors of enzymes in order to bind the enzyme via the cofactor to the carrier. Now he used tryptophanase and beta-tyrosinase. Tryptophanase will split tryptophan into indole, ammonia, and pyruvic acid. Beta-tyrosinase will do a similar reaction with tyrosine. It will yield phenol, pyruvic acid, and ammonia. And it is an enzyme that requires pyridoxal phosphate. Now their idea was to bind pyridoxal phosphate to a carrier, to Cephadec. This triangle is pyridoxal phosphate. And then attach tryptophanase via one of its places. It contains 4 subunits. Via one of its places for pyridoxal phosphate to the carrier, and thus have just one point to attach the enzyme, and in a very specific way, and the same thing with tyrosinase. Now, the various derivatives of pyridoxal phosphate that they have synthesized, I'll show you in the coming slide. Next slide. Well, this is, of course, pyridoxal phosphate, and the best product that they used was a 6-oxopyridoxal phosphate derivative. Of course, the aldehyde group will go to bind a lysine group. And this is bound here. This is a derivative of para-aminobenzoic acid, hexamethylenediamine, and this is the Cephadex or Cepharose. This is one type of bonding. They can bind via a bromoacetyl derivatives of Cephadex to the hydroxyl of pyridoxal phosphate, and they can bind the bromoacetyl to the nitrogen to form a pyridinium bond. Now, all these derivatives, once they are here, they will bind tryptophanase and they will bind tyrosinase in a very, very specific way. You can afterwards reduce the bond with sodium borohydride. Next slide. Now this is Mosbach has used other cofactors which are extremely interesting and important. These were adenine nucleotides that he bound to carriers in order to bind to them enzymes like lactic acid dehydrogenase, malic dehydrogenase, which require some of these nucleotides. Now, as you see, here is adenosine monophosphate, which has been bound here to cephatates via this side arm, and this is, of course, the PNH, which anyhow this dinucleotide, which is a coenzyme, of course, in reducing reactions, which it's again an adenine nucleotide derivative, which they again bind to this cephatate. You'll see a more complex reactions in the coming picture. Next slide. Well, here you see rather complex binding reactions, and he has synthesized really these purines derivatives in a very clever way indeed, which he finally binds to cephatate. Now, as a result of this, he has cofactors. This is, of course, cyclic AMP, which he can bind to the various carriers. Now, all these mononucleotides or cofactors that have been used here, they are very good materials for affinity chromatography, but they are very good materials to bind the enzyme. Now, it's rather very important to know that there are two types of cofactors. Cofactors that are not being utilized during the enzymic reaction, then the enzyme sticks. Coenzymes that are utilized, then you have to find out a method to renew the cofactor in order not to lose it, because usually it's an expensive material. Next slide. But then comes the major problem that I want to indicate here rather in a drastic way. When you bind such enzymes to various carriers, is the enzyme more stable or less stable? Now, to our enormous surprise, on many occasions when you bind an enzyme, it becomes much more stable than free enzyme. And I have here a few rather new examples. This is tryptophanase that we have seen its stability with temperature to split tryptophan, and as you see, it will lose most of its activity at 70 degrees. If you bind it at 70 degrees, it will still contain about 50 to 60% of its activity. This is the same for this beta-tyrosinase. You see at 70 degrees, the soluble enzyme has no activity. The immobilized enzyme, by these techniques that we have seen, has still 50-60% activity. Next slide. Now, it's rather interesting that towards guanidine, the stability rises enormously in this case. If you try to denature the enzyme, this is for tyrosinase. Now, with guanidine, native enzyme, it will be denatured practically at 2.5 molar guanidine. But you see at 2.5 molar guanidine, the immobilized enzyme is activated, and it will lose activity only at 5 molar. So we know by now that at least in some cases, and as you'll see, these are very many cases, one can stabilize enzymes by binding. Now, the Japanese, just because of this stabilization, and this tyrosinase and tryptophanase, just to use an example, as I've mentioned, tryptophanase splits tryptophan to indole, ammonia, and pyruvic acid. But it's a reversible reaction. If you take pyruvic acid and you add ammonia and indole, it will produce tryptophan. And of course, they made a column of this immobilized tryptophanase, and it's by now the best method available to synthesize tryptophan, by pouring pyruvic acid, ammonia, and indole. It's the best method to make tyrosine, by pouring phenol, pyruvic acid, and ammonia. And of course, by the same technique, they are making now DOPA, which is of considerable medical interest. But the question that of course, the biologists and biophysicists should ask, why are enzymes stabilized when they are bound? And unfortunately, I'm afraid that we still don't know the answer. And as long as there will be no basic work in this field, I'm afraid we will not know. And it will be a trial and error experiment. But perhaps it's worthwhile to mention that as a result of this stabilization, now 6-aminopenicillinic acid is produced in Beecham and in Bayer company in Germany with immobilized penicillin amidase that now a pilot plant is now running in order to transform glucose into fructose, glucose isomerase, which will keep on for months its activity when it is bound to glass. So the fact that they are stable is of enormous importance. But why are they stable? We still don't know. The simplest idea, of course, is that when you bind the enzyme to carrier, you simply freeze the vibrations of the macromolecule and thus perhaps inhibit denaturation or make denaturation more difficult. But it's certainly a most intriguing program. Next few slides will perhaps indicate some of the work of, well, here, by the way, this is tyrosinase or tryptophanase. If you make a column and pass through it tyrosine, of course, slowly the pyridoxal phosphate, which is free, will be eluted, and the bound enzyme will lose slowly activity. But if you add again pyridoxal phosphate, it will recover all activity. Next slide. Here are a few other striking examples of the stability of these bound enzymes. This is trypsin stability and activity as a function of pH. Trypsin has no activity at pH 9. These are various immobilized trypsin. As you see, they will still be active at pH 9 and 10. Next slide. This is the activity of catalase, which is also rather remarkable. If you take catalase in solution, it will lose activity within a couple of days and will not split any more hydrogen peroxide. But if you bind it with protein or with glutaraldehyde, it will be much more stable at 37 degrees. But if you carry out this experiment at 4 degrees, next slide, you see again catalase at 4 degrees within 35 days will lose all its activity, but when it is bound, it's completely stable at 4 degrees. So you stabilize these enzymes. Next slide. Well, this is a rather also interesting example of stability which will lead to what I'm driving at this freezing of conformation. If you take chymotrypsin and measure its activity towards acetyltyrosine ethyl ester at different temperatures, it will rapidly lose activity. At 50 degrees, it will have no activity. If you have it bound, it will go up in activity and then only at a later temperature will go down. Now, if you carry out another rather striking experiment, you heat the enzyme for 10-15 minutes at the various temperatures here, and then cool it again. So if it is immobilized, if you heat it to 60 degrees and then cool it to room temperature, it will still possess about 60-70% of its activity. But if you do this with a soluble enzyme, it has no activity whatsoever anymore. Next slide. Now, in order to test the conformational changes, together with Dr. Gabel and Itzhak Steinberg, we carried out a rather sophisticated type of an experiment. And I'll explain it rather very briefly. The idea was to bind chymotrypsin to Cefadex and to measure the fluorescence from the surface and to see whether there are changes in fluorescence. And let me explain the experiment. This is the cell. It's a quartz cell. You can put in particles with Cephalodex to which chymotrypsin has been bound. This is filter. You can filter off the medium and then resuspend your particles. Here you can inject whatever you want through this hole. You can measure the temperature with a thermistor. Now you put in here a beam of light and you measure fluorescence coming out from the surface. Now by this we excited it with 218 nanometers and measured the fluorescence, of course, of tryptophan. Now in order to see that the method works, you'll see in the next slide. If you take beta-alanine tryptophan and you illuminate it with 280 nanometers, you'll get out the emission spectrum 1, which is the emission spectrum in solution of beta-alanine tryptophan. If you bind beta-alanine tryptophan to Cephalodex and you carry out now this surface type of fluorescence experiment, this is the emission spectrum. So you see it's all right, so the method works. So now we started to look at the fluorescence of the tryptophan in bound chymotrypsin. Next slide. And already something striking we observed, that if you start to heat the bound enzyme to higher temperatures and you measure the intensity, it will go down, but if you cool it, the intensity of emission fluorescence doesn't come back. It stays kind of in a different shape. This is the shift in the maximum of fluorescence. If you heat to 60 degrees, then cool, there is no change kind of in conformation. Next slide. But then, this is the experiment which I'm driving at. And it's a rather interesting experiment. If you add to chymotrypsin toluidine sulfonic acid, toluidine sulfonic acid will fluoresce when it's bound to chymotrypsin. It's bound somewhere in a region which will increase the quantum yield of the toluidine sulfonic acid. But now if you add to the toluidine sulfonic acid bound enzyme free phenylpropionic acid, which goes to the active site, so the fluorescence will go down. And the idea is that free phenylpropionic acid goes to the active site of the enzyme. There is conformational change, and as a result of this, the milieu around the fluorescent dye changes, and the quantum yield goes down. So we have an enzyme where the fluorescent group is bound somewhere here, and the active site is here. And when you add the free propionic acid, there is conformational change, and of course the fluorescence of this place goes down in this case. But what happens if you take immobilized enzyme? It will bind the same toluidine sulfonic acid. This will be the intensity of fluorescence. Now you add free phenylpropionic acid, it will bind free phenylpropionic acid just the same, but there is no change in fluorescence. And our idea is that the enzyme is frozen, and it's much more difficult to make any movement, and therefore maybe that it leads to a more stable conformation. Let me have the lights, please. So this is perhaps one problem in enzyme engineering. But of course, once you have such stable materials, you can make columns, and it's the joy of the mathematicians by now to play around with columns and membranes and to calculate the rates of reaction. And of course, to our enormous satisfaction, work that we have begun with Rachel Goldman has been now picked up by most likely the best enzyme kineticists alive, who in Australia is publishing most likely every month another long mathematical paper in which he discusses the kinetics of immobilized enzymes. And of course, he has to take into consideration diffusion. This is perhaps the most characteristic feature in such reactions. And perhaps in order not to frighten you, I'll just show very briefly some of the very simple reactions that one can use in mathematical terms to explain the mode of action of such columns or membranes, or taking diffusion into consideration. But I won't go through all the mathematics, that it's only good for those who enjoy it. Now, next slide. Well, here is, of course, the formula that we have worked out for the mode of action of enzyme columns, and it's just a hint for the biochemists and the enzymologists. This is the well-known Michaelis equation. Change of substrate with time is proportional to enzyme concentration, Michaelis constant, and substrate concentration. If you integrate it, this is, of course, your result. T is time. Now, if you have a column, so time of a reaction is simply the height of the column divided by the linear velocity of flow. This gives you the time at which the substrate spends in the column. So this is the concentration of substrate at the top of the column. This is at the bottom. Of course, this is the concentration of product. And of course, it's the same equation in a column, but time is substituted by length of column divided by linear velocity. Next slide. But as I have mentioned, of course, diffusion is here of great importance. And if you run into a steady state, of course, you have the diffusion, the fixed law into any element of volume, and this is the reaction, by enzymic reaction, by which the substrate disappears. And of course, if it's a steady state, it's zero. At any other time, it's ds over dt. And the same thing holds, next slide, for the product. Of course, again, fixed law, and the product is formed by the disappearance of substrate. This, again, a steady state. And the solution of these equations is, of course, dependent on the boundary condition. And thanks God, there are many boundary conditions, so thanks God, there are very many solutions to this problem. May I have the lights? Now, let me just add that by now we have membranes with enzymes, and a lot of really fascinating work on these enzyme membranes has been carried out, mainly, I think, from the theoretical point of view. We now understand what will happen when acids are formed within these membranes as a result of the reaction. Just last month, and a superb paper has appeared, experimental as well as theoretical, to explain what will happen in such a membrane when hydrogen ions are liberated by the enzyme within the membrane. And I understand that Roy, who is here, Roy Kaplan, has even found conditions when there will be oscillations in product formation within such membranes, which is certainly of very great interest indeed. Now, let me just add finally one topic that I think may be of very great importance from the diagnostic point of view. And this has something to do with bioelectrodes. And the idea was, as we all know, we are daily using hydrogen electrodes, sometimes potassium electrodes, sometimes ammonium electrodes. But of course, the biologist wants to measure specific materials, their concentration, their amounts, changes in activity. And now one of the trends, which I think is a rather interesting trend, is to have a combined electrode. And let me explain the principle in a very, very simple way. The principle is based on the assumption, and I mention it because just last week I got Chemical News, and on the front page, there was a picture of a bioelectrode that and a rather interesting article on these bioelectrodes in this chemical news. So let me just explain what is the principle in a few words. The principle is a very simple one, that if I have, for example, an electrode which will measure ammonia, so ammonia can penetrate, we can measure activity of the ammonia by whatever system one has. It's certainly not a very specific type of a membrane. But now if I want to determine the concentration of urea any place, so how to do it? So the idea is, let's put here a little membrane or a little gel in which you have urease. Now if I dip this electrode into a solution which has urea, so the urea will come in, the urease will transform the urea, of course, into ammonia and carbon dioxide, and of course the ammonia or bicarbonate, and the ammonia penetrates in and you measure the concentration of the ammonia. So you can do it with ammonia gas and ammonium. You can now measure glucose by using glucose oxidase as an enzyme. Glucose oxidase will oxidize glucose, so it picks up oxygen. So if you have an oxygen electrode, you can measure by this glucose. And if you want to determine amygdalin somewhere, and you have an enzyme which will split amygdalin and form cyanide, you can easily transform cyanide into iodine. So if you have an electrode to measure iodine, so you directly measure amygdalin. And now you can, in principle, if you want, you can measure anything if you have a specific enzyme to attack it and to liberate the right ion for which you have an adequate electrode. Now, unfortunately, till now, these enzymes are not very stable in these membranes, so you have to prepare the membrane repeatedly, but it's certainly a very promising trend. Now let me therefore try and sum up what I wanted to bring to your attention. I felt that it is perhaps appropriate, while recalling the work of Weizmann, to indicate that at a certain period of his life, Weizmann spent a lot of time on fermentation problems, and was one of the first to introduce new fermentation techniques into industry. This work, which led to many important products, in his case butanol and acetone, afterwards led to many other products on a large scale. This was during the First World War. The Second World War saw a new progress in fermentation, in which materials which are not energetic, of interest from the energetic point of view, vitamins, antibiotics are formed, led to the new branch. Now is enzyme engineering, in which we will utilize enzymes instead of living bacteria, sometimes even dead bacteria, as a complex, as a part of an enzyme complex in order to carry out these reactions. And as in all such cases, I think those who have summarized the progress at this stage say so nicely and rightly that once one has a bright idea, it's so easy to write on paper all the various branchings of this idea, but it's so difficult to carry it out experimentally, and mainly when economic factors are involved. But it seems to me that at least in some cases, enzyme engineering has made serious progress has went into at least some industries, and from what I hear, it seems that it will enter within the near future into other industries and maybe also into the clinic. Let me summarize by saying again how moved I am with the death of Dr. Bergman. Practically everything that is in this Weizmann Institute here has taken place in its planning, in carrying it out. He has been the most wonderful man to his students and encouraging his collaborators. You'll find them now in any laboratory in Israel and in many laboratories all over the world. Let him rest in peace and let his memory serve to all of us as a symbol of a man dedicated to science and his country.
אני רוצה להודות לך אפרים בשם כל המכון וכל השומעים כאן על הרצאתך וגם על דבריך האחרונים. לפני שנתפזר רציתי רק להגיד לכם ידיעה שקיבלתי עכשיו ביחס לסדרי הלוויה לפרופסור ברגמן. למרות רצוננו להתחיל אותה מכאן, ייתכן שיהיו קשיים גדולים ולא נוכל לקיים זאת. ההלוויה אם כן תתקיים מחר תתחיל ב3 באוניברסיטה העברית. אני מצפה לנוכחות מרבית של אנשי המכון. תודה.